Hello and welcome back to the Crash Course Guide on Foundation Block. This is part 3 and today we're going to be taking a look at some more of the subjects from Foundation System. So first we're going to take a look at cell division, including mitosis and meiosis, and then we're going to take a look at some common bacteria and viruses and how they work. So let's start with cell division. It's important to understand why cells divide, and it's for growth, repair and reproduction. And there are two main types of cell division to be aware of. So first we have mitosis, and this is the production of two new cells that are identical to each other and the parent cell. So they're pretty much replicas of the parent. On the other hand, you have meiosis, and this is production of male and female gametes, so the production of four haploid cells that are very different from each other and the parent cell. So what's important is to link these types of cell division to their function. So for example, mitosis is absolutely excellent for repair and growth. So when something is damaged and you want to replace it with the exact same cell, mitosis is fantastic, and meiosis is brilliant for reproduction. So they both have their strengths. So the cell cycle is really important to understand, and it might be something you've come across before. So within mitosis, I'm sure you'll be aware that there's 4 stages of PMAT, so prophase, metaphase, anaphase and telophase. But what goes on outside of those and after cytokinesis, where your cytoplasm divides? Well, we have G1, S, G2 and G0. So the cell cycle is this series of events, and joining together G1, S and G2 makes up interphase, which is our stage just before we undergo mitosis. So interphase is our preparation stage. And then we also have G0, which is our non-dividing inactive state for the cell. It's not dead and the cell's not destroyed, but it's still there, but it's under cell cycle arrest. It can't do anything, it's just almost sat out inactive at the side. So the general process of how things go is interphase, mitosis, cytokinesis, and then it starts again. So the G1 phase. This is where cell contents are duplicated, but not chromosomes. In the S phase, all 46 chromosomes are duplicated by the cell. In the G2 phase, the cell checks for any errors and makes any repairs that are necessary. And then of course in mitosis we see cell division in the 4 stages of PMAT. So this is exactly the same picture, just in a little bit of different view, and we've now added something on called checkpoints. So there are 3 important checkpoints to be aware of within the cell cycle, and these are stages where we control and check what's going on in the cell cycle and make sure everything's working correctly. So the first one is the G1/S checkpoint, and this is at the end of G1, and it checks for nutrients, growth factors and DNA damage. In other words, the commitment to cell division point if the environment is favourable. The next checkpoint is at G2, and it's actually in the middle of G2, and this checks for cell size and DNA replication, and it checks all the chromosomes have been replicated correctly, which would have occurred in S phase, and if an error is detected here, the cell cycle is halted and the error is corrected before proceeding. And the whole idea of these checkpoints is to prevent mistakes being made, to prevent mutations. The last checkpoint is the M checkpoint, and this checkpoint is at the end of metaphase or the start of anaphase, and it checks that sister chromatids are correctly attached to the spindle fibres, and the cell will only continue with division if the spindles are all correctly attached. Now this may not make much sense to you now, but when we look at mitosis in a little bit more detail, hopefully it will. Now these checkpoints are regulated by cyclins, and cyclins help control the cell cycle. Cyclins are just a family of proteins that control the cell cycle progression by activating things we call CDKs, so cyclin-dependent kinase enzymes. And these enzymes are really important in order to allow the checkpoints to happen and make sure through each phase that the cell cycle is correctly regulated and controlled. So first let's take a look at mitosis. So remember, mitosis is the production of 2 new cells which are identical to each other and the parent cell. And mitosis, therefore, is the division of somatic cells, in other words, our non-reproducing cells. And the 2 daughter cells will be identical. So mitosis will only occur following interphase and when the cells are ready to divide, and it's made up of 4 stages followed by cytokinesis. And remember, cytokinesis is the splitting of the cytoplasm at the end. The 4 stages are prophase, metaphase, anaphase and telophase, and can be remembered by this PMAT. So mitosis. For normal cells, so DNA is present in an extended uncondensed form called chromatin. So cells can only replicate and transcribe DNA when it's in the extended state. So in order to prepare for division, the DNA molecules condenses to form chromosomes prior to division, and each chromosome is a single molecule of DNA. And easier to sort and organize replicated DNA into daughter cells. So stage 1, prophase. Chromosomes condense and spindle fibers form. So these are specialized microtubules that radiate from the centrioles. So centrioles is just your end of your cell, and they form these spindle fibers, which are almost like strings, which are going to attach to the centromeres, in other words, the middles of the chromosome. And it's important to be aware of how this looks both in a diagram like this, but also in a diagram like this. And chromosomes are captured by the spindles. So in metaphase, they all line up. The chromosomes line up along the equator of the cell, with one chromosome facing each pole. So you can see a chromosome here and a chromosome here, and they're facing each pole. And that's replicated here, how you can see the spindle fibers are attaching to the middle points, which are called our centromeres. Next we have anaphase. So this is where the sister chromatids separate, and they're pulled apart by the spindle fibers attached to the centrioles, shorten and pull the chromatids towards the pole. In other words, we're pulling a chromatid, half of a chromosome, towards each end of the cell. And lastly we have telophase, and this is where they're properly separated now, so spindle fibers disintegrate and the nuclear envelopes form around both groups of chromosomes. Be aware that the cytoplasm has not divided at this point. The chromosomes will revert to their extended state now, and eventually we have cytokinesis, which is the splitting of this cytoplasm, and a ring of microtubules contract and pinch the cell in half, and therefore we have two new diploids, in other words, full number of cells, identical cells are formed. Next we have meiosis. So this process is slightly more complicated, but very similar in what happens. So this is where a single germ cell divides into 4 unique daughter cells. So we have a parent cell at the top. The chromosomes make identical copies of themselves. They pair, and this time instead of having one division, as you do with mitosis, you now have 2. And this leaves you with 4 haploid cells, but actually all of these haploid cells are different. So when we say haploid, we mean a cell has got 23 chromosomes instead of the normal number 46. So sexual reproduction uses meiosis, and it's the fusion of 2 gametes to produce a single zygote. And it introduces greater genetic variation and allows genetic recombination. And what we mean by that is we produce offspring that look different. We don't all produce offspring that look the same, and if we have more than one child, they will look different. So fertilization essentially is the combination of 2 haploid gametes, so 23 plus 23 to equal the normal diploid number of 46. So in meiosis there are 2 divisions of the nucleus, meiosis 1 and meiosis 2. So in meiosis 1 we have the same kind of thing happening. We have prophase, metaphase, anaphase and telophase. Now 2 things happen really here which are different to what you see in mitosis. The first one is crossing over occurs, and essentially this is the crossing over of the chromatids in order to introduce genetic variation. And we also have independent assortment, and this is how the chromosomes line up in the middle of the cell along the equator, and it's different. So then when we pull them to opposite poles, we may get bits of chromosomes joined together being pulled to opposite ends of the cell. If this doesn't make too much sense, it'll come clearer in a second. When we understand meiosis 2, and the same thing happens again, and we end up with 4 cells which are all haploid in nature. So meiosis is all about creating genetic variation, and the 2 ways we do this, as we've already said, are crossing over and independent assortment. So crossing over occurs during prophase 1. So homologues break at identical locations and then rejoin with a different chromosome, and this creates new combinations of the alleles on each chromosome. For revision of what an allele and a chromosome are, go back to video 1 in this series. So this process occurs randomly several times on every chromosome. The other process that instills genetic variation is independent assortment, and this occurs during metaphase 1. And alleles for one trait separate independently from alleles for another trait, and each allele combination is equally likely to occur. So thinking about cells, bacteria are prokaryotic microorganisms, and animal and human cells are eukaryotic. and be aware of the differences between prokaryotic and eukaryotic cells, what they contain, what they look like, and the key differences, for example, that prokaryotic cells have a flagellum and eukaryotic cells do not, and so on. Also be aware of the functions of what each of these aspects of the cell do. So for example, what happens in the mitochondria, what does the Golgi body do, and what's the purpose of having a smooth and rough endoplasmic reticulum. So bacteria have a variety of morphology, and what that means is they look different. So there's coccus, rod or bacillus, and spirillum, and these are the different ways that bacteria appear and look. So the cell walls are different in bacteria. You can have gram-positive bacteria and you can have gram-negative. The easy way to remember this is that gram-positive bacteria will stain purple when you do the crystal violet staining test, and gram-negative bacteria will stain red or pink. So the difference in stain colour is due to gram-positive bacteria retaining the crystal violet stain because of its thick peptidoglycan wall. So in the wall of gram-positive organisms, it's got a thick peptidoglycan layer, and this retains or keeps hold of the crystal violet stain, causing it to stain purple. The gram-negative bacteria do not have this, and as a result, it cannot retain that crystal violet stain, and will therefore appear red or pink. So bacteria grow by binary fission, and this is an absolute rapid process. We have the lag phase, where the bacteria start to grow. We have the log or exponential phase, where the population doubles every 20 minutes, and then we have the stationary phase, where the growth stops, and finally we have death, so bacteria die faster than they multiply. We'll cover lots more on bacteria as we go into other videos in this series, and also through second year when we learn about specific bacteria and the diseases that they can cause. Next, let's take a look at viruses, and viruses are specific, and they're made up of a nucleic acid genome, a protein capsid, and viruses, remember, are only able to multiply by infiltrating a human cell and hijacking its machinery to allow it to replicate. Viruses don't have any machinery themselves to allow replication, and therefore they must enter into a human cell and almost hijack its machinery to allow it to replicate within. So thinking about viruses, we can classify the infection. So classification makes it possible to make predictions about the properties of a virus, i.e. its replication, its structure, its pathogenesis and transmission, and this is particularly useful when discovering new viruses. So hopefully this is familiar to you. So it's kingdom, phylum, class, order, family, genus, species, and this is the way we can classify organisms. This up here just shows you an example of how we can classify different types of viral infection. So let's take a little bit more of a closer look at that. So A, for example, is an acute infection. It happens once, it happens quick, and then it's over. For example, influenza. B is a latent infection, in other words herpes, so you have one bad infection first and it may reactivate several times individually in the future. C is a progressive condition, so for example HIV, and here we can see it occurs and gradually continues or gets worse over time. And lastly we have chronic infection, and this is where we have an initial acute infection and it just remains. It's persistent for good, and an example of this is hepatitis B. And that's everything for this video. Thank you very much for watching. I hope you found it useful. If you do have any questions or feedback, please feel free to get in touch and let me know. Thank you.